Good afternoon, students. So in my earlier video, I have discussed the basic PCR technique that has been developed by Professor Carey Mullis. And for this discovery or the invention of this process of polymerase chain reaction, he has been awarded with the Nobel Prize. Now, in this current video, I would like to discuss in depth with different other PCR tools that are being utilized or used or performed by different laboratories. So it's Dr. Rajib Majumder from Department of Biotechnology, Adamas University. So what is a PCR? So PCR is basically the polymerase chain reaction by which we prepare several copies of a particular DNA. So it's basically called the molecular photocopying. The polymerase chain reaction is a first and inexpensive technique used to amplify or copy several small fragments of the DNA. Because the significant amount of the sample of the DNA are necessary for different downstream application in the molecular biology, recombinant DNA technology, and in genetic analysis. So the isolate the piece of the DNA are nearby impossible without amplifying them through PCR. So that's why different components are used, as I mentioned in my earlier video also. Like we need a template DNA, we need different primers, we need DNA polymerase enzyme that will polymerize. For that, we need to add DNTPs. We need to add specific buffer with some cofactors. We need magnesium chloride for optimization of the PCR experiment. How does it work basically? So we all know that DNA is actually a supercoiled double helical structure. So for that, what we need to do, we have to apply heat to separate the two strands of the DNA. Now, when the two strands of the DNA are separated in the process called denaturation, then we can add the primers. Denaturation step have been executed at temperature above 95 degrees Celsius. In the present slide, you can actually visualize, you can actually see that upon application of the heat, the supercoiled DNA structure is separated into two strands of the DNA. Then, in the next step is called the annealing, while we actually anneal or add the primers at a specific complementary region of the DNA. Now, as a rule of thumb is that we need to set the annealing temperature at least 4 to 5 degrees Celsius less than the lowest Tm calculated of either these primers. That means if the least Tm or melting temperature has been calculated of one of the primers is 55 degrees Celsius, so then as a rule of thumb, we can set the annealing temperature at 50 degrees Celsius to 51 degrees Celsius. Now, the primers cannot extend until we add the building blocks, that is the DNTPs, and DNTPs cannot join automatically until we add a specific polymerase enzyme, that is Taq DNA polymerase. or the DNA polymerase have been isolated from Thermus aquaticus. Now, once we all add this in a reaction vessel, so the Taq DNA polymerase will keep on adding and keeping lots of copies of the same DNA. But the recombinant DNA technology of molecular biology is not restricted to only to this conventional PCR. Rather, there are several other PCR techniques that have been done by several laboratories, several researchers. So the first one that I would like to info or that I'd like to convey my message or I'd like to discuss now why different other PCR procedure or process than the conventional PCR process has got so much importance. It's all that suppose you have designed a primer against a suitable region of the gene of interest that you want to amplify. So by checking the sequence, you are actually preparing your primers, your forward primers, your reverse primers. But if the primers would not anneal only to the specific region, rather, if the primers will go and bind to several other regions along with your specific region, so what happens? So the Taq DNA polymerase will actually amplify lots other position wherever your primers will go and bind, resulting in a non-specific amplification of the gene, and we will be getting different PCR products, some specific and some non-specific bands. So the aim is to minimize the appearance of non-specific PCR product by improvising this basic PCR technique. So the first technique that I would like to mention is the hot start PCR. Now, what is the purpose? Now, in these two uh, uh, images, you can see there is a conventional PCR in the left-hand side, and there is a hot start PCR shown in the right-hand side. You can see that to prevent the non-specific PCR product to appear and also to prevent the formation of the primer dimers, prior the actual cycle will start, the Taq DNA polymerase can be added to the reaction mix already at 95 or 95 degrees Celsius in the hot start PCR. So the difference is that the enzyme, the Taq DNA, the polymerase that we actually use, it's actually a hot start Taq DNA polymerase. It is not a conventional Taq DNA polymerase. Rather, these enzymes remain inactive in ambient temperature, and only it becomes activated only when the temperature will rise above that temperature. So the hot start Taq DNA polymerase, the hot start Taq DNA polymerase are either enzyme linked or oligonucleotide linked or chemically modified in such a way to keep them inactive at the ambient temperature. However, when the temperature will rise above 94 degrees Celsius, an actual cycle will start only when this hot start Taq DNA polymerase will become activated, and then it will amplify your gene of interest through several copies. In this way, we can minimize the formation of non-specific PCR product, and also we can prevent the formation of primer dimers. In another PCR process, we call the reverse transcription polymerase chain reaction. What is that? Now, when we have a genetic material other than DNA, if it is RNA, for example, during the COVID period, we came to know about the virus that is SARS-CoV-2, the COVID virus, which is the RNA virus. So we know that RNA is a single-stranded molecule, and a single-stranded molecule, we cannot actually involve them in a PCR directly. So before that, this RNA molecule has to be converted into its complementary DNA by the enzyme called reverse transcriptase. And the process called the reverse transcription, the reverse transcriptase enzyme, actually convert this RNA molecule into the complementary DNA or cDNA. Now, once we'll actually prepare the cDNA, that is the first strand cDNA synthesis is done, then by taking it as a template, the second strand is formed, and as a result, we supposed to get the double-stranded DNA from one single-stranded RNA. Now, when we actually get this double-stranded DNA now from the RNA, cDNA, we can now follow the ideal PCR procedure. So RT-PCR is basically a laboratory technique combining the reverse transcription of the RNA into the complementary DNA and amplification of the specific DNA target using a polymerase chain reaction. It is primarily used to measure the amount of the specific RNA because as much the RNA is produced, that RNA will be converted into the cDNA, and as much as the RNA is converted into the cDNA, we can have an amplified product of that. So it is primarily used to measure the amount of the RNA present. This is achieved by monitoring the amplification reaction, sometimes using a fluorescence probe, and then if it is a real-time monitoring of how many number of the cycle is being conducted, how many amount of the DNA has been produced. So using the fluorescence probe in a real-time way. So this process is called the real-time PCR. And also when we actually need to quantify this, that what is the actual amount, it's basically the quantitative PCR. Now, sometimes when we actually combine this quantitative PCR with the RT-PCR routinely, we can actually achieve the analysis of the gene expression, and also we can do the quantification of the viral RNA in research and different kinds of the clinical settings. We call qRT-PCR or quantitative RT-PCR, which is one of the biggest and one of the most important PCR experiment in present scenario. Next, when we actually do this real-time reverse transcription PCR, that is conversion of the RNA into the cDNA, we can actually do this into two different procedures. One is called the one-step RT-PCR. One is called the two-step RT-PCR. Now, in one-step RT-PCR, where we actually do only one experiment, where we add our RNA, reverse transcription enzyme, my buffer, my all different PCR mixture, everything in one shot. The advantage is that it's a very time saving. It actually saves a lot of your time. But the limitation is that as there is no in-between quality check of which type of complementary DNA has been synthesized, and on the basis of the complementary DNA, we can actually achieve to get the PCR product. So if there is some error in the synthesis of the complementary DNA, so this error will remain in all the next amplification through the PCR, and the error will keep on going in all the several downstream copying or amplification. So to overcome this process, to overcome this problem, we can also perform a two-step RT-PCR where in the step 1, we actually add our RNA, reverse transcriptase enzyme, oligo DT primers, DNTPs, buffer, and other mixture, master mix, and other, to finally get the complementary DNA, and we can have a internal quality check of the first 10 cDNA that have been synthesized in the first step 1. And once we are sure that, yes, my cDNA, the cDNA that has been synthesized is good enough, it's pure, and uh, it can actually, we can continue with the cDNA in step 2, where we add our complementary DNA now, along with my DNA polymerase, my PCR primers, my DNTPs, buffer, etc., to get several PCR products from the RNA. And we can quantify through QRT-PCR and qPCR. So that means in the two-step RT-PCR, the first step is the reverse transcription carried out for the first in one reaction to get the cDNA, and in the second step, we do the PCR using that cDNA carrying out in the second reaction or second vessel to get the PCR product. Now, next in your screen appears, it's a colony PCR. It's a colony PCR. Now, sometimes what happens that when you are directly targeting, or you are actually a microbiologist and you are to do some kind of metagenomics kind of that, and you have got a lot of colonies of the bacteria. Now, if there are several number of the different colonies of the different bacteria, now it is almost impossible to task that to maintain each and every bacterial colony into pure culture form, and then to further and to prepare different media for individual colonies so that the bacteria will grow, and then and perform several different experiments for extraction of the genomic DNA from whole different types of the bacteria, and then do PCR one by one. So it will actually take a lot of time, and it's very, very time consuming. So that's why we have another interesting PCR technique. We call it a colony PCR, where the bacterial colonies have been picked and added to the PCR reaction, where the reaction already has the master mix. But for that, you know what? The PCR requires a template DNA. Now, if the bacteria, if you are adding the colonies from the plate, so the colonies means the live a population of the live bacteria present in a colony. So bacteria has to lyse, and bacteria of the cell, so that the DNA will come out. So that's why we need to add the lysis buffer to lyse the bacterial cell from the colony. So the two sets of the primer amplify two different plasmid DNA, the insert specific primer. Now here we actually use two different primers. One is the insert specific primer, another one is the insert specific flanking region specific primers in the right hand side. So two sets of primer are used to different plasmid DNA. One is the insert specific primers that amplify the gene of interest, whereas the vector specific primer will amplify the flanking region along with the insert. In this way, we can actually minimize, because you know what, the DNA of the bacteria is very long, the plasmid is very large in size. So if we can narrow down our location of the DNA, if we can narrow down in such a way that, first of all, using the flanking region specific primer, we are minimizing that part, we are narrowing down. And finally, when we have the narrow down, that is in the yellow, this yellow stretch, not the gray stretch, the yellow stretch. And then using a insert specific primer, we can get our desired PCR product and just we can overcome in this way the appearance of several non-specific other primers product or PCR product. Then we can do a DNA sequencing to identify our insert whether it is correct or not. So colony PCR is a very interesting technique uh, in nowadays where you have several bacteria from the metagenome sample, and in a plate there are several colonies. It is very difficult to maintain each and every colony into the pure culture, and then finally we do a colony PCR for that. Next is basically the touchdown PCR. Next is basically the touchdown PCR. What is the unique about that? It is a method for increasing the specificity of the PCR reaction. And in the touchdown reaction, the annealing temperature that we set, that will gradually down by 1 degree at a time. So if we are not sure that at what annealing temperature that we need to fix to get a desired PCR product, we can actually follow the touchdown PCR where we first fix a range of the temperature, suppose from 66 to 55 degrees Celsius, and in every cycle, there is a gradual decrease in the annealing temperature. You can see in the gradient that is 66, 65, 64, like this, and it will go down up to 55 degrees Celsius. So the touchdown PCR uses a cycling program where the annealing temperature is gradually reduced at least 1 to 2 degree per second cycle. The initial annealing temperature should be several degrees above the estimated TM of the primer. And in this way, we can reduce, in this way, we can reduce the appearance of non-specific PCR product formation. Because if the primer is not annealed properly, we cannot get our desired PCR product desired. And if the primer will go and bind to several other non-specific region of the DNA also, then again we will get to see the different non-specific primers, PCR product. So that's why fixing or finding out the actual and best annealing temperature for my PCR reaction is one of the most crucial part to do a PCR, a foolproof PCR. Next is basically the nested PCR. So nested PCR is again another PCR technique, very useful, where we actually using this nested PCR also, we first of all, we minimize or narrow down our gene of interest by removing other non-specific DNA portion of the template DNA so that my primer have to bind or primer will bind only to the specific region and not to the other region. Now that's why suppose my DNA is very large, and if within the DNA there is a specific gene, DNA of interest of the gene where I need to anneal my primers, but if there in the DNA other portion of the DNA have the complementary sequence also, my primer may bind to the other location. So that's why if it is so desirable is that if I can somehow narrow down the entire gene by removing other part of the template DNA, only keeping a narrow position, a narrow DNA segment, so we can actually increase the chance of binding the PCR primer only to the specific region. And if the PCR primer will actually bind to the specific region only when we can get our specific PCR product and no other product non-specifically. Now, finally, we are coming to a most important PCR technique nowadays that have been utilized for identification of the various strains of COVID virus, SARS-CoV-2, Omicron, Delta, in one single shot. When you need to do a cohort study that if there are 100 number of the people suffering from the COVID-19, or I mean the SARS-CoV-2 virus, COVID virus, and we know that at this moment there are several strains of the COVID virus exist in the universe or in. So that's why out of 100 number of the people, all are suffering from the COVID. If we need to find out that which patient is suffering from which strains or variant of the COVID virus, then the multiplex PCR would be the most suitable. Here, one of the most important application is the detection of the various infective microbes present in the surface. So the various strains of the SARS-CoV-2 have been detected using this approach in recent days. In a single conventional PCR reaction can encounter a single uh, uh, nuclear polymorphism and consequently identify only a couple of genotypes. Whereas using this multiplex PCR approach, many SNPs can be ruled out in single reaction. This makes possible if we study the many genotype at once. In the image, you can see that suppose in the SARS-CoV-2 there are 4 different variants, and we are actually designing the primers against 4 different variants, provided the gene sequence of all these 4 have been already identified. Okay, and finally, in one single experiment, we are actually adding all the 4 primers designed against 4 different variants. We are adding all the reagents for the PCR. And finally, if we try to run an gel electrophoresis, you can actually find the different products. So that's why multiplex PCR is one of the most suitable and important PCR technique by which in one single shot, we can actually study many genotype, or we can actually detect several variant strains of one specific type of the microbe in from environmental sample or patient sample. So here I am actually concluding that these are the whatever I have discussed. These are the different other PCR approach or PCR techniques other than the conventional PCR that are very useful for this.